Hello and welcome back to OmicsLogic Data Science. In this program, we discuss methods for analysis of big data and data wrangling. These are essential skills for bioinformaticians, as well as those interested in other types of non-biology related research using various methods of data analysis. So what we do in this program is we want to try to understand how to work with different types of omics data, performing exploratory analysis and the steps to process big data and prepare it for downstream analysis steps, then try some of the big data analysis tools for large-scale project data and apply different types of machine learning techniques and learn about the important links between biological data and the actual biology that it represents to master biological interpretation. So in the first part of the program, we focused on some of these introductory steps, how to work with different types of omics data like genomics, transcriptomics, metagenomics, and others. We saw some of the challenges associated with preparing that big data for analysis, and we did an overview of different methods to handle and structure preparing the data for visualization and in-depth analysis. And so in the second part of the program, we are now focusing on a variety of different methods, including univariate and multivariate analysis methods, as well as examples of how to apply machine learning and artificial intelligence to the workflows that we would like to perform with such data. So in today's session, we will continue to speak about unsupervised learning methods, and that includes dimensionality reduction and clustering. So specifically, we will discuss how several different approaches to clustering, such as k-means, hierarchical clustering, and different types of spectral clustering called DBSCAN, can be applied to different types of data and to different types of questions. And we will also talk about how to practically apply them using the T-Bioinfo platform, as well as using R and Python. And so then we will speak about some of the challenges for various methods and when to apply which algorithm, how to validate your findings, how to use these techniques and how to combine them together to achieve your analysis objectives. So a lot of interesting topics that we want to cover today. Let's get started. So before we jump into the topic of today's session, let's do a little review, kind of remember where we are in the program, what we have covered so far, and where are we going? Why are we performing these steps of clustering and dimensionality reduction? So far, you should have learned about preparing the data for analysis, exploring it, and linking patterns that have been found with biological interpretation. What does it mean? And how to get an intuition about what you are expecting to find. To do that, in the first part, we discussed a lot of the analytical pipelines that can load in the raw sequencing data and prepare a table of elements that could be analyzed with the different methods that we discussed. So these pipelines vary by the type. So here, for example, you see a pipeline for transcriptomic data analysis, or here you see a pipeline for metagenomic data analysis. The pipelines are different, but they utilize some of the same principles. And the principles are pre-processing, or clearing up some of the noise from the raw sequences, mapping, how do we actually align it to some reference, either genome or a whole maybe database of elements, and then how do we compare different conditions by quantifying how much of different types of elements do we actually have. And that can be a table of expression where we quantify mRNA for genes, mutations, or maybe operational taxonomic units. So how do we actually get those taxonomic units and how do we interpret that data? For example, in the case of genomic data analysis, we can obtain a table of somatic scores per sample across all of the genomic positions in the DNA of the reference organism. High somatic scores correspond to frequently observed mutations in sequencing reads, and we interpret them to mean that these are mutations that were acquired as a result of cell division. That's why they're called somatic mutations. For example, we can talk about transcriptomic data. We have a table of continuous numeric data for gene isoform or exon expression levels, annotated by gene ID and organized consistently across samples in a given experiment. So in this table, you have samples as columns, gene IDs as rows, and the intersection is the level of expression of that gene for that sample. In the case of metagenomic data, we look at operational taxonomic unit. So 16S ribosomal RNA amplicon, which means that these are only regions that are amplified, metagenomic sequencing data. We have a table of OTUs, or operational taxonomic units, also across samples, with proportionate abundance in each microbial community. So together with this table, what we need is the annotation of the taxonomy of these operational taxonomic units to interpret the composition of the microbiome sample. So to explore these tables, we also learn how to visualize different properties of the data and summarize them using the statistical properties that can help us explore the major signal and variation across samples. We also learned to use different statistical approaches or analysis techniques that help us find specific patterns of interest, such as, for example, differentially expressed genes between groups of samples. The statistical methods that we talk about are used widely today to find patterns that are sort of easy to interpret biologically. So, for example, hypothesis testing for each gene that represents genes that are consistently and strongly different between conditions. So differential gene expression analysis is commonly understood to represent a biologically meaningful way to identify key genes that could be interpreted and validated. And so finally, we spoke about some of the major challenges because of data variability, and how the steps to explore data are very important to differentiate between signal and noise. Specifically, in the statistical approaches, we oftentimes look for the most significant patterns, and then we also have this challenge of minor patterns that could actually describe data that covers many different conditions across a lot of variability and in noisy data. And that is when we turn to machine learning. And so machine learning methods can be separated into two main categories of methods. Unsupervised machine learning that focuses on detection of patterns in data. And the second main category is the supervised machine learning methods. And these methods rely on availability of training data, so data that has been labeled. Once a data set of labeled data is accumulated, it can be used to train a model that will be able to apply this knowledge to analyze new data, for example, by predicting a class. So with unsupervised machine learning methods, we start with a data set. Typically, it's a table of elements, just like what we saw. And here, we can use shapes as examples. So we can use a clustering method, for example, hierarchical clustering or k-means clustering. And these could identify groups in the data. And the fact that it places each sample into a group based on some feature, in this case shape, is the learning process. And so the outcome of the learning method is identification and assignment of each sample to a group, which means that each group has a boundary and we can study each group separately. One unsupervised machine learning method that we already talked about is principal component analysis. Let's imagine some kind of a data set that looks like a cloud. We can find a particular type of a projection. For example, we can take two of the first features that can either show us low variance or high variance. Now, in either case, maybe the features that we have in our data don't represent the full data in a very accurate way. So what we can do is we can have a projection based on principal components that are projected in this original data that maximize variance or explain variance of the original dataset. Principal component analysis uses explained variance to show us the most accurate way to represent our data, but it is not always going to work well for any kind of data. In metagenomic sequencing data, we're interested to understand how diversity changes across samples and can be used to understand composition, which can be done using non-metric multidimensional scaling, or NMDS. The goal of NMDS is to collapse information from multiple communities, sites, etc. into just a few, so that they can be visualized and compared. Unlike other ordination techniques that rely on primarily Euclidean distances, such as principal coordinates analysis, NMDS uses rank orders, and thus it is an extremely flexible technique that can accommodate a variety of different kinds of data. Another method that seeks to deal with various assumptions about data distribution and complex structure is TSNE, or T-SNE, which stands for T-Distributed Stochastic Neighbor Embedding. It uses connectivity to establish neighborhoods in data and then shows a lower dimension representation of the connectedness between data points using a projection mapping that seeks to accurately represent the original topology in our dataset. Perplexity is what T-SNE would use to represent the distance between samples, which is a measure for information that is defined as 2 to the power of the Shannon entropy. In T-SNE, the perplexity may be viewed as a knob that sets the number of effective nearest neighbors. It is comparable with the number of nearest neighbors, k, that is employed in many manifold learning. So what we will do today is we will continue our discussion about machine learning, and we will turn to clustering. Cluster analysis, or clustering, is the task of grouping a set of objects in such a way that objects in the same group are more similar to each other than to those in other groups. Importantly, this approach tends to be deterministic. It seeks to determine for each point which group to assign it to. While this is not the case for all methods, some determine belonging to a group based on initiation, which can lead to different interpretations each run. This is the main difference between simply representing the data in a lower dimension and assigning an actual cluster to each point. And this is specifically helpful when some overlap between point groups make it difficult to accurately determine belonging to a group, and provides a rationale to study cluster points as a part of a single unit. So to explain, let's take a look at our data. Here we have known groups that are clinical subtypes of breast cancer. Each gene has a profile. So for example, this is a profile of this particular gene. This is a small example, so we only have 15 genes. But if we plot a bar plot for each gene, we start seeing some patterns, and they show some similarity between samples of the same type. Some logic behind similarity can now be determined by automatically assigning each sample into a defined group so that it belongs to just one cluster using all of the gene expression values. So here we can see the patterns, but how do we use all of this data together and actually come up with all of the patterns determining the same thing, or belonging of samples to a particular group? There are many different types of clustering, and they are defined by different clustering algorithms that use several different approaches. So let's take a look at some of these. The partitioning algorithms divide data objects into a number of partitions, where each partition represents a cluster. These clusters should fulfill the following requirement. 1, each group must contain at least one object, and 2, each object must belong to exactly one group. For example, we have the k-means algorithm, where the center is the average of all points, and coordinates represent the arithmetic mean. There are many other partitioning algorithms, such as k-modes, PAM, Clara, Clarons, and FCM. The second example is the hierarchical based, where data is organized in a hierarchical manner depending on the medium of proximity. Proximities are obtained by the intermediate nodes. A dendrogram represents the datasets, where individual data is represented by leaf nodes. The initial cluster gradually divides into several clusters as the hierarchy continues. Hierarchical clustering methods can be agglomerative, or bottom-up, and divisive, or top-down. An agglomerative clustering starts with one object for each cluster and recursively merges two or more of the most appropriate clusters. A divisive cluster starts with the dataset as one cluster and recursively splits the most appropriate cluster. The process continues until a stopping criterion is reached, frequently the requested number k of clusters. The hierarchical method has a major drawback, though, which relates to the fact that once a step, merge, or split is performed, it cannot be undone. Some examples for hierarchical clustering include birch, cure, rock, and chameleon. Density-based. Here, data objects are separated based on their regions of density, connectivity, and boundary. They are closely related to the point nearest neighbors. A cluster, defined as a connected dense component, grows in any direction that density leads to. Therefore, density-based algorithms are capable of discovering clusters of arbitrary shapes. And also, this provides a natural protection against outliers. So the overall density of a point is analyzed to determine the functions of a dataset that influence a particular data point. DBSCAN, Optics, DBCLAST, and DENCLU are algorithms that use such a method to filter out noise, which are outlier, and discover clusters of arbitrary shapes. Another approach is grid-based, where the space of the data objects is divided into grids. The main advantage of this approach is that it has fast processing time because it goes through the dataset once to compute the statistical values of the grid. The accumulated grid data make grid-based clustering techniques independent of the number of data objects that employ a uniform grid to collect regional statistical data, and then perform the clustering on the grid instead of the database directly. The performance of a grid-based method depends on the size of the grid, which is usually much less than the size of the actual dataset. However, for highly irregular data distributions, using a single uniform grid may not be sufficient to obtain the required clustering quality or fulfill the time requirement. Some example methods include wave cluster and sting that belong to this category. So finally, we have the model-based approaches. These are methods that optimize the fit between the given data and some predefined mathematical model. This is based on the assumption that the data is generated by a mixture of underlying probability distributions. And also, it leads to a way of automatically determining the number of clusters based on standard statistics, taking noise, which are the outliers, into account, and thus yielding a robust clustering method. There are two major approaches that are based on the model-based method: statistical and neural network approaches. MCLUST is probably the best known model-based algorithm, but there are other good algorithms such as EM, expectation maximization, which uses a mixture density model, conceptual clustering such as Cobweb, and neural network approaches such as self-organizing feature maps. So let's take a look at several examples for the first three types of clustering methods that we described so far. Hierarchical clustering is one of the most popular approaches because it allows clustering on different levels. Even though we typically choose and have to specify the number of expected clusters k, the way this algorithm works does not actually require the k, but only is needed at the end, once the hierarchy of data points is established. To see how, let's demonstrate how this method works. So let's say we have our samples organized in some space. So imagine a PCA plot, like this one, 2D plot, where each sample is positioned in some perspective that best represents the data variance. The way the algorithm works is it finds points in this space that are the closest to each other and assigns the center for those 2 points as a pseudo cluster. We perform the same operation to find the nearest neighbor and organize them together. And so when we connect those 2 points, that's called linkage. And we use different methods of measuring distance between points to establish the link. And ultimately, the algorithm will stop once we reach all of the samples belonging to the same cluster. So this was an example of bottom-up agglomerative clustering. Let's see how this algorithm actually works. So here we have our points distributed in this two-dimensional space. You can see that the nearest points are organized as a single point, and once all of the points reach the same cluster, now we know that we have built the full hierarchy. Now, based on this hierarchy, we can determine some kind of a threshold where we cut off this hierarchical tree and determine the number of samples in each cluster. To run clustering on the T-Bioinfo platform, you will need to upload the table of expression that we generated when we analyzed, for example, our cell line data. This data can be downloaded directly from the lesson or from a demo pipeline in the cell line RNA-seq demo. Clustering methods are located under the data mining section of the platform. Hierarchical clustering is an example of agglomerative or bottom-up clustering method that starts with attributing each sample to a separate cluster. It then finds the pair of the most similar samples and unites them in one cluster that is further distributed as one quasi-sample. This procedure is repeated until coming to one cluster that contains all of the samples. So when we put the data into the pipeline, it will load the data, perform clustering, and put out the result in several different formats that we will discuss in a little bit. So what are the parameters that we have to choose? First of all, we have to choose the right linkage, which is a parameter that determines the definition for measuring intercluster distance. Single sets the definition as the distance between the 2 nearest points in 2 clusters, whereas complete defines the distance as that between the 2 farthest points. The 2 Ward methods are variation of Ward's minimum variance method. The distance parameter is the second one, which specifies the distance metric to use in measuring distance between data points. And this could be Euclidean, Manhattan, maximum, or Canberra. And then we have the cluster's expected count, which is a parameter that sets the cutoff height of the hierarchical dendrogram to the one that results in a given number of clusters. And then we have the option to transpose our data, which is a parameter that allows us to transpose the table and either analyze genes or analyze samples. So what is cluster linkage? So there are several different types we talked about, right? We talked about the average linkage, and this is the method that finds all possible pairwise distances for points belonging to two different clusters, and then calculates the average. This method is less affected by outliers than the other ones. Single linkage, which uses the minimum difference to determine clustering. The distance between two groups is defined as the distance between their two closest members. And we have other types like centroid and complete linkage. Then we have different parameters for distance, and understanding the choice between all of these different methods really is based on your understanding of the input data. So the hierarchical clustering algorithm is a process by which a computer uses the input file, where columns are objects and rows are features. Then it assigns each object to separate cluster or group, and then it evaluates all pairwise distances between clusters using a chosen distance metric, and constructs a distance matrix using the distance values. Then it finds the pair of clusters with the shortest distance and merges them into a single cluster. And then it repeats these steps until we end up with a single cluster for all of the samples. And as a result, what do we get? We get the hierarchy of our samples organized into a dendrogram. Here we have an example of a dendrogram, and you can see that we can cut it off at different points to get the desired number of clusters. At the very top, we have 2 clusters, then we have 4 clusters, then we have 8 clusters. Another method that we will talk about is k-means, which is probably the most popular clustering algorithm of all. It is a partition type of an approach. The algorithm itself is relatively simple. Starting with a pre-specified number of cluster centers, each point is initially assigned to its nearest center. In the next step, for each segment, the centers are moved to the centroid of the clustered points. The points are then reassigned to their nearest center. The process is repeated until moving the centers derives little or no improvement, which is measured by the within cluster sum of squares, which is the total squared distance between each point and its clustered center. In the k-means pipeline, we also input a table of expression in a txt file, and after running the algorithm, we're able to observe 3 different things. First, the k-means clustering does not do a great job of clustering for a lot of these examples because of the many different features that we have, essentially thousands of features per sample. So if we compare the 2 methods, k-means and the hierarchical clustering, we will see that they produce different results. So essentially, because of the metrics that we select, we expect to find the same number of clusters, for example 4, but depending on the metric that we use, we can see those clusters separating, and we can always evaluate the results based on what we know about the samples. For example, we know which sample belongs to which clinical phenotype. Now, another type of clustering is spectral clustering. Spectral clustering techniques make use of the spectrum, or the eigenvalues, of the similarity matrix of the data to perform dimensionality reduction before clustering in fewer dimensions. The similarity matrix is provided as an input and consists of a quantitative assessment of the relative similarity of each pair of points in the dataset. The basic algorithm consists of several steps that are based on the Laplacian matrix, which is a way to represent a graph of interconnected nodes and study graph relationships. The same approach is used for nonlinear embedding or dimensionality reduction techniques like UMAP. Spectral clustering does not work well on all data because it is computationally expensive unless the graph is sparse and the similarity matrix can be efficiently constructed. A special case of spectral clustering is called DBSCAN, which stands for Density-Based Spatial Clustering of Applications with Noise. So now that we reviewed the methods and some of the ways that we can run them, Let's discuss when such methods can be used and how to interpret the results or analyze their outputs in a meaningful way. For example, we can compare how different clustering and dimensionality reduction techniques can provide us with insights about data. So if we label our data based on known labels, like here we have different types of breast cancer subtypes, we can see how the major structure of data is separated into two big groups. And we can see in our PCA scatter plot that we essentially get the same thing, right? Everything is separated between luminal and all the rest. So the most obvious way to use the threshold is to set the threshold to divide everything into two major types. The distance between them is determined by the height, and you can see that the distance in both our PCA plot and the cluster dendrogram could be comparable. A good way to test clustering models is to also compare phenotype data with the unsupervised classification we created. So in this case, we see here a smaller number of genes that was used, and as a result, we can also determine the most far separated groups that we have. The same thing goes for PCA, where we can see a separation between the luminal group and the non-malignant group right here. Oftentimes, clustering can also help us find outliers. For example, here, we can see an irregular tree that highlights the sample HCC202. And then if we want to actually check whether this makes some kind of a sense, we can see that actually this specific cell line, HCC202, has been studied and found not to belong to the same category of cell lines that we typically think it should be assigned to. Clustering is also widely used in data visualization because it helps us organize visual patterns into heat maps, for example. So we visually determine groups of samples and genes. And a lot of times this is going to be useful when we perform differential gene expression and we want to demonstrate how differentially expressed genes actually are organized into two different groups of samples, like we see here on the right. Or on the left, we can also identify specific common genes that are organized in some kind of a form between groups of samples. Another way to use clustering is also to find correlated expression of genes across samples that can reveal meaningful biological function that is shared between groups of genes and affected in similar ways through perturbation. So when we have different conditions, we expect those conditions to affect biological function. So if we take our data set and we separate it here in 4 clusters and then we visualize what are those patterns for each cluster, we can see that they are correlated. Now, if we find a group that is highly correlated and different from other patterns in the data, we can then assume perhaps they somehow are linked to each other and maybe they form a biological function. So going through annotation, as you can see here on the bottom right, can help us understand what could be that function. Okay, so now that we reviewed the methods and we understand how to run them, let's discuss how clustering methods can be compared, evaluated, and selected for a specific objective or job. One simple way to understand how well your clustering works is to try to plot a confusion matrix between known classes, if they are available, and the cluster assignment. In this way, you can see which sample is not being assigned to the proper category. If the annotation is not available, the confusion matrix can be used to see if the same sample is being assigned to an inappropriate cluster or to several different clusters. And that will result in maybe changing the settings or maybe iterative runs of the same algorithm. In some cases, your challenge would be to determine how many clusters to expect. In that case, you can use metrics like inertia for k-means or silhouette for a variety of different methods. Inertia measures how well a dataset was clustered by k-means. It is calculated by measuring the distance between each data point and its centroid, squaring the distance and summing these squares across one cluster. A good model is one with low inertia and a low number of clusters. In addition to the confusion matrix, a hierarchical clustering method can show a dendrogram of linkages between samples which can be interpretable and can improve your understanding of data. This can be done visually or with a metric score. like the silhouette score. The silhouette score is a method that is common for many types of clustering methods. The average silhouette method computes the average silhouette of observations for different values of k. The optimal number of clusters k is the one that maximizes the average silhouette over a range of possible values for k. The silhouette coefficient is calculated using the mean intra-cluster distance and the mean nearest cluster distance for each sample. Thus, you can choose k and iteratively calculate the score to maximize nearest cluster distance. The silhouette ranges from -1 to +1, where a high value indicates that the object is well matched to its own cluster and poorly matched to neighboring clusters. If most objects have a high value, then the clustering configuration is appropriate. If many points have a low or negative value, then the clustering configuration may have too many or too few clusters. The gap statistic is an approach that can be applied to any clustering method to evaluate how well we have assigned the number k. So what it does is it compares the total with intra-cluster variation for different values of k with their expected values under null reference distribution of the data. The estimate of the optimal clusters will be value that maximizes the gap statistic, which yields the largest gap statistic. So this means that the clustering structure is far away from the random uniform distribution of points, which means that you have chosen the correct number of k. In many cases, a visual representation of data can help significantly to understand why a certain group is determined to be a cluster, or why an outlier can reduce the quality of clustering. Combining your analysis with a representation of data can be a great way to mine it for patterns and discover ways to optimize the results of your analysis. A completely different challenge can be when you are trying to apply the same approach to feature similarity analysis. So for example, we want to understand not the clustering of samples like we see here, but we also want to try to understand what groups are formed by different features of our dataset. One way to do this is to understand the loading of features, or how features contribute to separation between groups of samples. In that case, we can identify the top features, and those features can have a direction in the same space that separates the clusters that we have. But when we want to look at the actual clusters in our data, we can assume that biological differences between groups of samples represent some kind of an influence, and we can study how that influence affects different genes, which, as we will see in a few minutes, reflects the differences between samples and can be useful for biological interpretation. Now let's discuss how to run and understand the coding practices behind some of these methods. On the T-Bioinfo platform, you are running methods implemented in R. For example, when running the k-means pipeline, you will notice the output file called rpack.zip. Inside the zip file, you will find the source code designed to take in the parameters from the platform and translate those into variables used in the main call of the clustering method. To understand the code and how to get more from each method, we provide a number of tutorials in the course Introduction to Data Science in R. The first place where you encounter clustering is in the PCA lesson. There you will find advanced implementation of PCA visualization with clustering methods Clara, Pam, and Fanny. The methods are well suited for high-dimensional data that is reduced using PCA and scaled. Clara stands for Clustering Large Applications, which is a method that works on large datasets as compared to k-means or k-medioids, and it will select random observations from the dataset and perform the partitioning. A similar method, called PAM, stands for Partitioning Around Medioids. Using the library ggfortify, you will learn to perform PCA and then utilize the stats package to cluster the PCA objects and produce cluster results in a visual format using ggplot2 autoplot function. In R, there are at least several packages that implement clustering algorithms in a user-friendly fashion. One of these is the stats package that covers many analysis tools for statistical analysis and clustering. hclust
is dedicated to hierarchical clustering using the agglomerative bottom-up approach with linkage and distance settings in the usage call. The same package implements k-means and other partition methods in R that are called in a very similar way to each other and are covered in the online tutorials. To practice, you will find examples in the clustering lesson. This will include running the analysis and visualizing results with a dendrogram. You can additionally explore interactive dendrogram options that can be used for coloring your dendrogram and finding the right number of clusters to select. Additional packages in R that you can consider are packages cluster and pvclust. Cluster is a package that implements hierarchical clustering with additional features like the agglomerative coefficient and other useful visualization tools. Another package, pvclust, adds a p-value for hierarchical clustering using bootstrap resampling. In Python, scikit-learn contains a number of methods for clustering data and evaluation of clustering performance. On the Learn portal, you will find tutorials to practice syntax, but also coding challenges, and a link to an interactive notebook where multiple methods of clustering are compared. The given assignment after clustering methods illustrate how to apply the same logic to clustering genes instead of samples. As a result, the additional notebook offered in the lesson explains the benefits of density-based clustering methods like DBSCAN to identify closely related genes that might share some biological function or can be used to identify artifacts in data or noise. In the follow-up exercise, you will see how these methods can be used after nonlinear dimensionality reduction, like tsne and UMAP, where the number of clusters can be more meaningful, but also the practice includes dealing with removing outliers and working on thorough comparison of results from similar but different methods of clustering. In conclusion, what did we learn in today's session? We reviewed unsupervised learning techniques like dimensionality reduction and clustering in detail. These include partition-based methods like k-means, hierarchical methods like hclust or birch, and density-based methods like DBSCAN. We also talked about the ways we can use clustering to identify differences between samples or genes and find meaningful patterns. We also spoke about ways to overcome challenges in clustering and make interpretation a bit easier. Finally, we discussed the ways that we can combine various methods of dimensionality reduction and clustering to find biologically meaningful patterns in complex data and how to use these methods on the T-Bioinfo platform or implement them in R and Python. Thank you for joining this session, and I hope to see you in our next session dedicated to classification.